But here is a little bit more of a focus on what are those individual amino acids doing in this in this catalysis. Now, what we've got and what we're going to focus on are this glutamic acid and this aspartic acid right here. Now, what is going to happen is this glutamic acid number 35, well, it's protonated, and it is actually going to function as an acid. Because what it's going to do is it is going to enable the immediate release of our part of our substrate. This proton goes to right here on our OH. What is left behind is an oxonium ion. So we have that carbocation there. Okay, so we've got a deprotonated glutamic acid 35 in our first step. Well, after our substrate binds. In that first step, though, we don't really have anything, we don't have that aspartic acid really doing anything in that very first step. All that's being done is glutamic acid donates a proton, and boom, it moves on. Well, aspartic acid is going to take over in that second step, and what's going to happen is that aspartic acid is going to serve as a great nucleophile, and our substrate is going to serve as a great electrophile, in which case we're going to form a covalent bond between our aspartic acid and our substrate. When that happens, then we can bring in a water molecule. That water is going to serve as an acid, and when serving as an acid, it's going to donate a proton to our glutamic acid, so our glutamic acid can be protonated again and returned to its original state. The remaining OH is going to go to our carbocation, and now all of a sudden we've got a carboxylic acid, no, we don't have a carboxylic acid. We have this OH group on what was previously a carbocation. So when it comes to what we were looking at earlier with the 18, the isotope of oxygen, that 18 isotope, well, it would have been introduced here, and tracking it there, well, that's where everything goes to where it originally, that's how it gets back to its original state. Now, the cool thing about this is this mechanism shows acid-base catalysis and covalent catalysis. And by showing that, what we see is, well, how dynamic different amino acids are. Now, what we did see is glutamic acid serving as an acid. Okay, aspartic acid, does it serve as a base? No, it doesn't. Aspartic acid serves as a nucleophile. Now, if you were to switch those amino acids and structure independent, do you think that glutamic acid has what it takes to function to form to act as a nucleophile? Absolutely. If you subbed glutamic acid and aspartic acid out for one another, I mean, they both have that carboxylic acid, and sure, they are different in terms of length, but they could both kind of substitute for one another. And you might see one mechanism down the road where glutamic acid serves as a nucleophile and aspartic acid serves as an acid in the same mechanism. So I think that this is a good demonstration of how amino acids are fairly dynamic. Now, an example of a kind of complicated or a pretty cool family of enzymes is our serine proteases. Serine proteases are a family of proteolytic enzymes that have a serine active, or an active site serine. Two examples are chymotrypsin and subtolysine. Thrombin is also a serine protease, and thrombin functions in blood clotting. So what it does is it cuts polypeptides, or it cuts proteins that ultimately are going to be factors in basically preventing, that will, I believe they prevent blood clotting. I can't remember exactly in that sequence. They enable blood clotting. Trypsin, chymotrypsin, and elastase are all digestive enzymes. They're going to be found in your gut, and they're going to help you digest proteins. Subtolysine is a bacterial protease, but these are all serine proteases. Plasmin is a protein which breaks down the fibrin polymers of blood clots, and tissue plasminogen activator, or TPA, cleaves the proenzyme plasminogen, yielding plasmin. Okay, now this suffix right here, OGEN, That's one thing that we're just about to get into with this concept right here, zymogens. So zymogens is a really fancy, fun word, and zymogens are activated by proteolytic cleavage. So a zymogen is an inactive protease. So there are zymogens are inactive forms of enzymes. In many cases, proteases are produced as zymogens. So we've got our example right here of chymotrypsinogen in our final phase or our final state of alpha chymotrypsin. So chymotrypsinogen, because it has that OGEN suffix, you should read that as that's the inactive form of an enzyme. Now, that enzyme is going to be produced, but it's not going to do anything until it is cleaved by another protease. So it is going to be cleaved by trypsin. Now what trypsin is going to do is basically, here's our primary amino acid sequence. Trypsin is going to break a peptide bond right there. Okay. Now what you also see in this sequence is this S to S. Those are disulfide bonds. So you can, in this case, you're going to break that amino acid sequence or that polypeptide bond, but the protein's going to remain kind of as a full thing because you have these disulfide bonds that are still intact. Okay, so trypsin is going to break peptide bonds, then to go from here to here, from pi chymotrypsin to alpha chymotrypsin. Well, pi chymotrypsin is going to cut itself, and it's going to, in the process, release a pair of dipeptides. So this auto activation, auto catalytic activity is going to release the entire or the complete active enzyme. So zymogens are basically inactive, but then something else sets in motion the activation of them. This is completely irreversible because you can't really remake a peptide bond. So this is a way in which an enzyme can be regulated. We'll talk about that when we talk about enzyme regulation, but this is just an example. So this right here is a depiction of 3 different serine proteases. And I'm going to talk about a mechanism for serine proteases in my next video, but this right here shows you the difference between the so-called specificity pockets, which is a pocket adjacent to the active site of serine proteases, for trypsin, chymotrypsin, and an elastase. Now trypsin, in its specificity pocket, has an aspartic acid. What that aspartic acid is going to do is it's going to seek out positively charged amino acid R groups. So something like arginine is a good example of a positively charged amino acid R group. Chymotrypsin has a fairly hydrophobic, H-phobic, specificity pocket, in which case it's going to look for kind of bulky residues. Elastase has a very small specificity pocket, in which case it's going to seek out only very small amino acid residues. So these different enzymes, these different proteases, are going to select the different peptides that they cut based on sequences. So if you gave something like RAD, a tripeptide like this, to trypsin, is it going to cut it? Yes, it is, because what trypsin is looking for is it is looking to cut after a positively charged residue. And so I'm going to switch to green here, and I would say that trypsin would cut right here after that arginine. Now then, if we gave this same polypeptide to chymotrypsin, well, does chymotrypsin, so chymotrypsin's looking for a hydrophobic residue, a bulky one like a tryptophan or maybe even a tyrosine, maybe. In this polypeptide right here, in this tripeptide, I don't see that. We don't have one of those. If you took this same polypeptide and gave it to elastase, would elastase be able to cut it? Yeah, chances are, because what elastase is looking for is it's looking for a small residue. So after that alanine, that elastase is probably going to be able to cut that. This all has to do with the specificity pocket, is what it's termed. So trypsin cleaves on the C termini of basic positively charged amino acids. Chymotrypsin cleaves on the C termini of aromatic residues, something like a tryptophan or maybe even a tyrosine. And then elastase is going to cleave on the C termini of small neutral amino acids. So these are all ways that these different serine proteases, or that these serine proteases distinguish themselves from one another. So that specificity pocket that I described earlier. Okay, so the mechanism of serine protease is something that I'm going to go into a little bit more in my next video, but one of the things that I want you to take away from it is basically every single serine protease has 3 residues that are absolutely essential. 1, serine. Next, a histidine, and finally, an aspartic acid. Now, what these different residues do, well, that's a little bit variable in terms of the mechanism. That aspartic acid, though, it's kind of interesting because it really only plays a structural role. All right, well, I hope this is helpful, and thank you very much. If you have any questions, please don't hesitate to email me. Have a good one.